The section from the VCAA study design we're going to look at now is the one called Biological Knowledge and Society. And specifically what we're looking at is techniques that apply DNA knowledge. So we recently were looking at DNA manipulation, which was looking at some different tools and techniques to manipulate DNA. This is more looking at how we take that information and our ability to manipulate DNA and what we know about DNA, and we use that to apply it in society in some way. So we're looking at gene cloning, genetic screening, DNA profiling, and we're also having a look at some of the social and ethical implications of those particular applications. We're also going to take a look at genetically modified organisms, transgenic organisms, because there is a slight difference between the two, and how we can use transgenic organisms and genetic modification in agriculture in particular to increase crop productivity by providing resistance to predation by insects, resistance to diseases, and we'll also look at some of the biological, social and ethical implications of those. Gene cloning is a method we can use to get multiple copies of a target gene or of a particular gene of interest. So if you think back to when we looked at DNA manipulation, another way we could get multiple copies of a gene or a section of DNA is through PCR, polymerase chain reaction. So this has the same principle, I suppose, in the sense that we're taking a certain section of DNA and we're wanting to make multiple copies of that section. But this time, instead of using a PCR machine, we're going to use bacterium themselves. So gene cloning, the way we would do it is, firstly, you would make a recombinant plasmid to insert the gene of interest into the recombinant plasmid. Then you would transfect some bacteria, so you place the plasmid back into a bacteria cell, and then we provide that bacteria cell with the right conditions for it to undergo binary fission, and we allow it to replicate itself many, many times. And of course, each time it undergoes one cycle of binary fission, we're taking one cell with the plasmid containing the gene of interest, and that's dividing now into two cells. So we've got two cells that have the gene of interest, and then we go 4, then we go 8, and so on and so forth. The other effective thing with gene cloning is not only are we getting another copy of the gene of interest each time one bacteria cell undergoes binary fission, but plasmids themselves, as you might recall, can also self-replicate. So you've got a bacteria cell which has the plasmid inside each individual bacteria cell replicating itself to make multiple copies, and then you've got the bacteria cell itself replicating to make even more copies. So what we're left with after a certain period of time is many bacteria cells that contain multiple plasmids that contain the gene of interest. So therefore we've got many copies of the gene of interest. What do we do then? Well, we can do two things. One is we can isolate those copies of the gene of interest, and we might want to do that so that we can conduct some research on the gene. We might also want to do that because we want to insert the genes into plants, might be a gene for pest resistance, or we might have wanted to insert that gene into the bacteria, and then we actually switch it on in the bacteria, so it changes that bacteria in a certain way, gives it some characteristics that were desirable. The other thing we can do is we can, probably along the same lines of what I was just talking about, we have the bacteria cells which have been cloned to have the gene of interest. We turn the gene of interest on so the bacteria cells start producing the protein that is coded for by the gene of interest. And then with that protein, we can conduct research on the protein, or we might actually extract the protein from the bacteria cells, and we can use them in humans, for example. So we can use bacteria as little factories to grow human growth hormone. We can use them as little factories to grow proteins involved in dissolving blood clots that are used in heart attack therapy. So we've sort of got this little collection of bacteria cells, all with the gene of interest, and we're using those cells as a factory to produce proteins which we're extracting and using in other organisms. Another way we can use our biological knowledge and apply it in society is through genetic testing. So with genetic testing, what you're doing is you're testing a person and you're looking for any defective alleles in their DNA that are related to a particular genetic disorder. So genetic testing falls under two main categories. We have what we call genetic screening, and then we have what we call genetic testing. And the biggest difference between the two is that genetic screening is looking at a population, so an organised genetic screening program that is offered to all members of a population, whether those members appear to be at risk or not. Whereas genetic testing is testing of an individual and screening an individual's DNA, most likely an at-risk individual, to see if they have any defective alleles related to a particular genetic disorder in their genetic makeup. Because we say genetic testing, we're looking at someone at risk, that means for some reason that person has been identified as having the potential to possess that defective allele, and they've either elected to get a genetic test themselves, or maybe they've been recommended by a doctor to get a genetic test to confirm or rule out if that defective allele is actually present. With genetic screening, and the purpose of offering it to a whole population, is early detection, or if it's not detection, it's early exclusion of the chance of that particular person having an inherited disorder. So you see in Australia genetic screening programs used with newborn babies, where they screen newborn babies for a range of different conditions such as cystic fibrosis or phenylketonuria, PKU. And the benefit of genetic screening is if an individual is identified as having a positive reading for that defective allele, well, at least you can have early intervention. And if it's not treatment, it can have, well, it's not cure, you can have early treatment. On the flip side, the problem with genetic screening, particularly because you're doing it to everyone in the population, not just those worried or deemed at being at risk, is the chance that the test does produce a false positive result, where it tells you you've got a condition when you actually don't. Obviously, the other thing is just coping with the fact that you've identified that your child does have a disorder. So things that we do to sort of eliminate that, or not eliminate it, but regulate it, control it, is you can only do genetic screening for genetic conditions where the benefits of doing the genetic screening outweigh any potential harm that would come from doing the genetic screening. And some measures we have in place to sort of make it the case that it is beneficial is that the screening program needs to relate to a significant genetic disorder, so one that's particularly common or one that has particularly harmful characteristics associated with it. Also, along with that, we need to make sure that whatever we're testing for, there is a suitable test out there that does have a really high degree of accuracy and a really low rate of recording false positive results. And finally, we can only do genetic screening for conditions where we do already have either a treatment or an intervention program that can be offered to any individuals that are come back with a positive result. So it sort of avoids the here's a genetic condition that your son or daughter might have, but at the moment we don't have any intervention or support programs we can give you. So it eliminates that. This slide outlines a few of the different types of genetic tests that somebody might get. So remember, genetic testing, we're talking about an individual, an at-risk individual, getting screened. So we could have a screening to identify if there's an increased risk of a particular disease. So some diseases, such as breast cancer or ovarian cancer, people with a particular mutation in a specific gene are said to have a higher risk of developing that disease. So it's not so much a defective allele that's causing a disease, but it's a defective allele that is related to an increased risk. So you could get an adult screen to identify if they've got that particular allele. We can do carrier testing. So if someone's a carrier, it means they can carry a defective allele, but in their phenotype, they're not actually expressing that allele. So they don't have a disorder themselves, but of course, if they're carrying a defective allele, there's a chance that they can pass that on to their offspring. So carrier testing is a test used to identify that. Prenatal screening and diagnosis is genetic testing on embryos or fetuses. So during pregnancy, there might be cause for concern that your fetus or your embryo might have a particular genetic condition. This increases with age quite often, so older mothers are often encouraged to get some sort of prenatal screening and diagnosis above and beyond that of what all babies get. So all babies get a blood test and an ultrasound screening, and then on top of that you can sort of get some other more detailed genetic screening which would identify or rule out any birth defects related to the child's genetics. Predictive or pre-symptomatic testing, as it suggests, is you've got a person that is not showing any symptoms of a disease yet, so a disease that would occur after birth or later in life, but you can get testing to see if you actually have the genetic makeup where you might develop that disease later in life. And then finally, we got embryo biopsy or pre-implantation genetic diagnosis. This is where we can apply genetic testing to the embryos used in IVF to determine whether or not that embryo has a defective allele, and that would of course influence whether you implant that embryo into the female or whether you discard that embryo and obviously implant one that does not have any defects. In society, genetic testing seems to pose as many positives as it does negatives, and you can have a bit of a read about the different positive and negatives here. But as a whole, in terms of the positives, you're pretty much looking at the positive being early detection. So early detection of the chance of developing a particular disease or having a particular disease, which means you can monitor your health a bit more effectively. Early detection that can help with decision making. So I suppose decision making in terms of pregnancy and having children, but also decision making in whether you're going to get what type of genetic testing you might get on your embryo or fetus during pregnancy. Another positive is it can rule out uncertainty. So if the test comes back negative for carrying a defective allele, I suppose you don't have to live your life constantly worried about the chance of what if you do carry it, so you know that you don't. But on the flip side, pretty much for all of those positives, you also have a negative. So most of our negatives are based around causing anxiety for the people that have been tested. So if you get a positive result for carrying a defective allele for a condition that appears later in life, well, from that time of your positive result, you know that there's a very likely chance that you'll be developing a particular genetic disorder later in life, and that could be, quite stressful and cause someone to be quite anxious thinking about that. Also, it can cause anxiety for parents in terms of decision making. So a result comes back showing that your embryo or fetus is positive for a particular genetic condition, and then you need to make those decisions based on continuing on with the pregnancy or terminating the pregnancy. There's always a chance that the results could be inconclusive, and then they come back and you're still left with that uncertainty of whether you have a defective allele or not. And then finally, there's some issues around confidentiality. If we're doing all of this genetic screening, this genetic testing, that data is being kept somewhere, and there would be some concerns about privacy and data like that getting leaked to health insurers, which could negatively impact on how people are treated. The next application we're going to look at is how we can use our knowledge of DNA to identify people, essentially. So when we're using DNA for identification, the most common application we refer to is in sort of like forensic science and using DNA evidence to link a person to a particular crime. You could also use it in terms of paternity testing and using DNA to identify the father of a child or something like that. So when it comes to using DNA in identification, we have two types of DNA that we use, and you guys will be aware that we have our nuclear DNA in our nucleus, which can be organised into chromosomes, and then we also have DNA in our mitochondria, which we call mitochondrial DNA. We can use both of these sources of DNA for identification purposes. With our nuclear DNA, it's inherited from both parents. It undergoes recombination, and within the nucleus, compared to mitochondrial DNA, we have a relatively small amount of nuclear DNA because we only have 2 copies in each cell. Mitochondrial DNA is a circular piece of DNA found in your mitochondria. It's not inherited by both parents. It's actually only inherited maternally. So your mitochondrial DNA was inherited from your mother. Her mitochondrial DNA was inherited from her mother. Her mother inherited hers from her mother, so on and so forth. So in terms of siblings, every sibling has the same mitochondrial DNA because each sibling inherited it from their mother. So if we're talking about DNA for identification, mitochondrial DNA can certainly narrow it down to a family, for example, but you can't narrow it down to an individual. Mitochondrial DNA does not undergo recombination, so any changes that do occur in mitochondrial DNA comes through mutation, whereas the changes that occur in your nuclear DNA can occur during meiosis when crossing over occurs to create recombinant chromosomes. With mitochondrial DNA, we have a lot of it. So it's in every somatic cell. We have multiple mitochondria in every somatic cell. So it's often, when we've got really old specimens, mitochondrial DNA is often what's left because it degrades more slowly than nuclear DNA as well. So, fossils, specimens, things like that, often we're finding mitochondrial DNA in them opposed to nuclear. If we're going to use DNA to identify different individuals, well, we need to look at a part of DNA that differs between each individual. And for the most part, DNA between individuals is very, very, very similar. So particularly in our coding regions, so our regions of DNA that code for proteins, obviously as humans, for us all to be human, we essentially all need to make the same proteins to function as a human. So it makes sense that our coding regions of DNA are all very much the same. Amongst our coding regions of DNA though, we do have non-coding regions. So non-coding regions of DNA are still sequences of nucleotides, but those sequences do not form genes that code for particular proteins. And what you'll find is that all DNA, whether it's nuclear or mitochondrial, in its non-coding regions, it'll have what we call hypervariable regions. And a hypervariable region, if you break that name down, hyper means a lot, variable meaning different. So hypervariable regions are particular regions of DNA that vary greatly between individuals. So if we're looking at nuclear DNA, what we're looking at is a particular hypervariable region, which we call a short tandem repeat, an STR. And by using a collection of different short tandem repeats. So a short tandem repeat is a repeated short sequence that forms a hypervariable region in someone's DNA. So typically, if you look at this example here, we've got CTA, 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 CTA. So it's a short tandem repeat. Short in the sense that it's only ever 2 to 5 base pairs in length. So that's the thing getting repeated. Tandem, meaning it's one after another, so it's repeated consecutively. And repeat obviously meaning repeat. So CTA, 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 CTA. So what we're saying is all humans, for example, they would have a region in their non-coding DNA where they all have this repeat of CTA, 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 CTA. But what differs between people is how many times it's actually repeated. So whereas I might have this sequence repeated 4 times, like the example, another person might have the sequence only repeated 2 times, and we could use that to identify that person from me. So we'll talk a lot more about short tandem repeats over the next few slides. So the hypervariable regions in nuclear DNA are short tandem repeats. The hypervariable regions in mitochondrial DNA, we just call them hypervariable regions. And there's a particular part of mitochondrial DNA called the D loop, and that's the sequence of nucleotides in mitochondrial DNA that seem to vary between people. They wouldn't vary a lot between siblings. Generally, family groups will have the same mitochondrial DNA unless random mutation has arisen, which it always does, but mutations take a very long time to occur. So the differences in hypervariable regions would be quite good to differentiate, say, one family line from another, but not so good to differentiate a brother from a sister, or a brother from a brother, sister from a sister. This next series of slides is possibly going to get pretty confusing for you, because using short tandem repeats and understanding how they're inherited and how they're used in identification, it can be a bit tricky to understand, and often it is, I think personally, better learnt using a whiteboard and your teacher sort of drawing it out. But that will happen in class as well. This will just give you a little taste of what's involved. So short tandem repeats, STRs, as I said before, they're in nuclear DNA, and specifically they're in the non-coding regions of nuclear DNA. Another name for an STR is a microsatellite, but typically in VCAA Biology we do use the word STR. It's a short repeating sequence, and the sequence being repeated is typically about 2 to 6 base pairs in length, and it can repeat up to 100 times. So in that example we looked at just before, that CTA repeated 4 times, that CTA could get repeated 100 times in a particular individual. We have different STRs. So an STR, you could think of it like a gene almost. So we have, in our body we use the word gene locus. So a gene locus is where a particular gene is located. So you'd have the gene locus for eye colour, gene locus for hair colour, gene locus for handedness, left or right. And then we also say that for every gene you have 2 alleles for every gene. It's the same as STRs. So you can have an STR locus. Let's say, for example, you have an STRA locus. Then you would have an STRB locus as well, an STRC locus. And typically, we, for a very long time, have used 9 different STR locuses to create a complete profile of an individual. So the complete set of STRs is unique to every individual except for identical twins. So if we're saying we're looking at 9 different STR locuses, what we're saying is, well, some people could be the same at some of those locuses, but no person other than identical twins have the exact same sequences at all of those 9 different STR locuses. So at an STR locus, as I said before, because we think of it as a gene, we have 2 copies of that STR locus in the form of alleles. So if you look at the diagram down here, we're saying we're looking at the STRA locus, and at that locus the short tandem repeat is GC, and in individual 1 he's got 1 allele for that locus, so that has GC repeated 1, 2, 3, 4, 5 times. It's a pretty dodgy looking 5. And then on his other allele he has that sequence repeated 1, 2 times. So at the STRA locus, individual 1, we would say his genotype is 5/2. If we compare that to individual 2, it's the same locus, so whenever it's the same locus, it's always going to be the same sequence. So what all individuals have in common is they all have the same STR locuses, and at each locus they all have the same sequence that's being repeated. The only thing that differs is how many times it's actually repeated. So we can see with individual 2 at this locus, GC is repeated 1, 2, 3 times, and on his other allele 1, 2, 3, 4 times. So his genotype would be 3, 4. And you can tell that those 2 individuals are different people. If we were applying some genetics terms to this, we would say person A has 2 different alleles because they have 2 different repeats. So we'd say that they're heterozygous at the STRA locus. Individual 2 also has 2 different alleles, so we would say he's also heterozygous at the STRA locus. If individual 2 had 3 repeats on one allele and 3 on the other, then we could say he's homozygous at that STR locus. This slide here talks a lot about what I was just explaining in the slide before, so I don't think I'll talk through it. I'll let you read through it to just give you another dose of what I was talking about. We might talk through the diagram at least though. So if we have a look at individual 1, this picture, let's split it down the middle. So individual 1, this diagram is showing us his genotype at the STRA locus, and it's also showing us his genotype at the STRB locus. So that's that one there, and the A one's up the top, and then it's doing the same for individual 2. Individual 1's STR number of repeats at the A locus is the same as the example we looked at before. So the repeated pattern is GC, and he's got 1, 2, 3, 4, 5, and 2. At the STRB locus, because it's a different locus, it is a different sequence of nucleotides, and that's what makes one locus different to another. So at the STRB locus, no longer is it GC getting repeated. The sequence that's repeated at the STRB locus is AGCT, and on one allele it's repeated twice, and on the other allele it's also repeated twice. So we could say individual 1 is heterozygous at STR locus A, but is homozygous, meaning 2 of the same alleles at STR locus B. Compared to individual 2, so he's got 3 repeats there, 4 repeats there. At STRB locus, he's got 1, 2, 3 repeats of AGCT and 2 repeats of AGCT in his second allele. See what I mean? Because it's the same locus, even though it's in 2 different people, it's still the same sequence getting repeated. The only thing that differs is how many times it repeats. What we can then do is we could take these STR locuses and we could isolate them and we could run them on gel electrophoresis. So if we look here for individual 1, this is showing us that his A5 allele, which is the first one, which has 5 repeats, spreads out to here. His A2 allele, which has 2 repeats, spreads out to here. And his B2 alleles spread out to the same area. Whereas individual 2, we've got the A4 allele placed here, the A3 allele placed here, because the A4 is longer than the A3, so it won't travel as far. The B3 allele here, because it's 3 repeats compared to the B2 allele, which was only 2, placed here, and we can sort them out. A good thing this could be used for is it's a nice diagrammatic to show you that individual 1 compared to individual 2, though they match up at one place here, for this to be the same person, they would need all of their fragments to be matching up. And the fact that they're not, this tells us very easily that it's 2 different people. If we were to run DNA from a crime scene, let's say in the middle well, and it banded out like this, then we would see that that's actually an exact match to individual 1, and then we could pretty confidently say that that's the same DNA and it's had to have come from the same person. So we use the word DNA profiling. So at the moment what we've talked about is what DNA do we use to identify individuals, and what specific part of the DNA do we use to identify individuals. And by using those sections of DNA that do differ between people, we can create a DNA profile of a person. So much like a profile of a person can sometimes say their height, their weight, their skin colour, their age, what they were wearing, a DNA profile doesn't tell you any of that information. What it tells you is what DNA they are carrying. So typically we would do it with the STRs. So a DNA profile of a person might be their 9 different STR locuses and how many repeats they have at each locus. So everybody except for identical twins does have a unique DNA profile, and what we would say is it's a 1 in a billion chance that 2 people would have the same DNA profile, which is really, really, really slim. So typically we say that each person has their unique DNA profile, except for identical twins. So if you're a dodgy identical twin, you could stitch up your identical twin pretty easily and you could frame them, but you shouldn't do that. That would be bad. So typically with the DNA profile, if I jump to our next slide, we have 2 different methods. So we can create a profile using restriction fragments. So this is called restriction fragment length polymorphisms, or we can create the profile using PCR and typically using STRs. So with a profile where we would use probes and restriction fragment length polymorphism, which is an older way of doing it, you would use restriction enzymes and apply them to a sample of DNA, and they would cut that sample of DNA up into different lengthed fragments, and then you could run that sample of DNA on electrophoresis gel, and it really works on the principle that the restriction enzymes would result in slightly different lengthed fragments of DNA when they're used to cut one person's DNA compared to another's. Profiling using PCR is a newer technique. So for example, you could take the STR region of a person's DNA and you could use PCR to amplify multiple copies of the STR regions, and then once again you can spread that on an electrophoresis gel and use that as a comparative sample against other samples that have been prepared in the same way. So essentially what we're doing in DNA profiling, comparing the base sequences of DNA from two or more individuals. To do that, we isolate the DNA, we amplify the DNA, and then we use the electrophoresis to sort the strands, and then for a visual comparison. You can do it with nuclear DNA and STRs, or you can use it with the mitochondrial DNA and their hypervariable regions in the D loop. And as I said before, often mitochondrial is used when we can't get nuclear DNA, but if you've got access to the nuclear DNA, that's probably the best sample to use each time, but you would go to the mitochondrial if that's not possible. Some problems with mitochondrial DNA is that it's inherited maternally. So as we said before, you can use it to sort down to a family group, but you can't use it to differentiate between siblings. How do we actually do DNA profiling? So here's our steps. So firstly, the DNA that we're going to use to create the profile needs to be isolated from each individual or each sample. So you've isolated your DNA, then you would use PCR not to create whole copies of the DNA, but just to create copies of the 9 different STR locuses present in each sample. So once you've used the PCR, you will have created multiple copies of STR-A locus for both people, STR-B locus for both people, STR-C, STR-D for example. Once you've done that, you load each sample into a different well. So typically you would use one well per different person. And for person 1, you're loading all of their DNA into that well. So all of their DNA from their 9 different STR locuses. For person 2, you're loading all of their DNA into the well next to it. And if you've got more than 2 people, you're obviously using more of the wells. You turn on your electrophoresis machine and they will then sort the fragments based on the length of the fragments. And as we know, the larger ones will travel less distance, whereas the smaller fragments will travel more distance. And because we're using STRs, and the thing that differentiates them is how many times the sequence is repeated, obviously the more times it's repeated, the longer that fragment will be. Here's a little example of how we might use DNA profiling and electrophoresis to do a paternity test. So what we've got here is we've got a mother, a child, and an alleged father. So what would have happened is DNA from the mother, the child, and the father would have been taken. Let's say PCR could have been carried out to get multiple copies of their different STR locuses, and then they've been run on an electrophoresis gel, and this is the results. Though we've got 1, 2, 3, 4 different columns here, the way this is supposed to be read is one big long line. So we have this first one on the left goes 1, 2, 3, 4, and then this next one would go straight after that, 5, 6, 7, 8, and this one would go continue on. Because your STRs are inherited genetically, that means you get one STR allele from your mother and you get one STR allele from your father. So when we're doing a paternity test, what we want to look at is the child should have their STR allele matching up with either the mother or the father. So wherever because we know the mother's the mother, it's pretty hard to not know the mother of a child. Because we know the mother is definitely the mother of the child, each time her band does not match up with the child, that child's band should match up with the father. And you can see here that that's a match with the alleged father. You can see here the child doesn't match with the father, but the child matches with the mother. Same deal here. Here he matches with the father. Matches pretty much with the father again here. Matches with the father down here. So matches with the mother here. When you're looking at this one here, you can pretty much say that each time the child's fragment length is not the same as the mum, it is the same as the alleged father. So it'd be a pretty good chance that the alleged father is actually the father of this child. With the ability to create a DNA profile of a person, not only can we use that to link a person to a crime scene, we can actually use that to create a DNA data bank. So much like a real bank that holds money, a DNA data bank is a bank of the different DNA profiles of people that have been convicted of serious crimes, and also DNA that's been found at crime scenes. So we actually have this national database that can be searched whenever DNA is found at a crime scene, so you can search it and see if there's a match on the DNA data bank. So we can use that to link people to the scene of a crime. You could link one crime to another crime, obviously use that to clear innocent people, and DNA evidence is really strong evidence in convicting a person of being guilty. How does it work? So you have your DNA stored in the database of all of these different peoples that have been, offenders before, and you would have, so let's say if we look at this picture here, there's the DNA profile of a individual that has been stored on the data bank, and we can assume that this is, say, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 10, let's say 10 different STR locuses, and then a gender marker. And here we've got 15, we've got 18, so you could treat that as one allele at this locus, another allele at this locus, and that is what the DNA profile would look like. We collect and determine the DNA sequence of the DNA found at a crime scene, and then you plug that into the data bank and you run it and you see if you get a match. And in this case, you can see that for every sequence down here, we've got a perfect match up here. So we would say that is a full match between the DNA found at the crime scene and that particular individual that's had his DNA stored on the database. So a full match is when all the numbers match up, and also the gender marker, so XY means male, whereas XX means female, the gender marker matches up as well. On this bottom one, you can see we've got the DNA profile of the individual, so it's the same individual, but this time we've got DNA found at the crime scene, and we've run that DNA, and you can see that some numbers match up, lots of them do, and the gender markers, but you've got 1, 2, 3, 4, 5 areas where they don't match. So we don't have a full match, we have a partial match. So a partial match occurs when some data points are missing from the crime scene. So you can see it's not that this is a different number to the individual in the data bank, it's that this sequence of DNA is missing altogether. And that could have happened because it may have been degraded or it might have been removed from the crime scene somehow. So you've got a partial match, which I suppose in the court of law is not as strong as a full match, but it's still pretty strong evidence that it is the same person. Finally, what you could do is you could do a search for family members. So sometimes where police have found an offender's DNA but that offender is not actually in the data bank, so they know the sequence of the DNA but they can't get a name for it, they might do a search to see if that offender has any close matches that indicate a family member or a close relative that they do have in the data bank, and then obviously they can go and start their search with that relative to try and get closer to this person that is responsible for the DNA that they've found. Some of the issues associated with DNA profiling are listed here. So with having DNA data banks in themselves, I suppose some of the concerns are whose DNA profiles should be added, so what severity of crime needs to be committed to warrant a person having their DNA stored on the data bank. And then also, should the DNA be stored on the data bank permanently, or is there a certain period of time where that person doesn't commit any more crimes where they should have their DNA removed? With familial searching, so that was that act of having the DNA from the crime scene but not having a direct match of who the person is in the bank, but then using the bank to search for close relatives, because the bank will give you a list of close relatives ranked in order from most closely related to least closely related, but those relatives would need to be in the data bank. A problem with that is a concern that investigators might rely too much on that sort of genetic lead to trace down a family line at the expense of other lines of investigation. I suppose the main concern, though, is are people who have their names in the DNA data bank, even though they haven't committed this particular crime but are just related to a person that has committed that crime, are they being unfairly discriminated against? When you compare it to, there could be many people that commit a pretty serious crime but they don't have relatives in the data bank, so that person's relatives don't get taken into account for the investigation. Finally, predictive testing, so that DNA that we collect from a crime scene, you could use that DNA to predict certain aspects of the person's phenotype. So, for example, you could say, well, based on this DNA we've collected, we believe this person will be, blue eyes, maybe of this racial background, etc., etc. Problem with that is how reliable is it to predict a person's phenotypes? Because we all know phenotype, yes, it is determined by a person's genotype, their genetic makeup, but it's also influenced by a lot of environmental factors. And also, does it reinforce some prejudice against certain groups in the community by promoting racial stereotypes that are associated with crime? Our next stop on the DNA knowledge and application in society journey is looking at this idea of manipulating genes within an organism, and what we call that is genetic engineering. So if you genetically engineer an organism, or you have an organism that's been genetically engineered, that means that their DNA has been altered in a particular way. And we'll have a chat in a moment about the different ways we can genetically modify or genetically engineer an organism. Why would we bother with genetically engineering an organism? And the point is because we want that organism to have certain characteristics or some desirable traits. Breeding organisms or manipulating things so that organisms display desirable traits is not a new thing, and it's something we have been doing for a very long time, more through selective breeding or conventional breeding. But in comparison to genetic engineering, there's certain benefits that you would get from genetic engineering that you wouldn't get from conventional breeding or selective breeding. So with genetic engineering, you can take DNA from almost any species and you can integrate that DNA into a target organism. The fact that DNA is universal is the thing that enables us to do that. Whereas with conventional breeding, you're only taking the genes from two very closely related species, and then you're getting two parents to breed together so that their offspring can show the desirable characteristics. In terms of time, it takes a lot of generations for you to achieve a phenotype in an organism that is what you want it to be. Whereas with genetic engineering, you can alter an organism directly, so it can happen in a much shorter amount of time. Also with genetic engineering, it's very specific, so you can take a single gene and transfer that into an organism, whereas conventional breeding, you're taking the entire genome from two different individuals and putting that together. So yes, you'll be getting the desirable trait, but you're also getting other traits that come along with that. Whereas with genetic engineering, you're getting that specific desirable trait in that organism, and the organism has all of its other genome the same as what it was before you integrated that gene. The probability of achieving the desirable outcome is higher with genetic engineering. And also with genetic engineering, not only can you transfer genes into other organisms, you can also silence genes. So you could take an organism, manipulate its DNA in a way that turns a gene off, whereas with conventional breeding, you can't actually do that. GMOs is a pretty common thing you might have heard of, which is genetically modified organisms. And then we have another word we talk about, which are transgenic organisms. And they're a little bit different in a way. They're sort of the same but different. So a genetically modified organism is an organism that has had its genetic makeup artificially changed. And that change can be in a number of different ways. Typically, it may have had a gene added into its genome or a segment of DNA added into its genome that's come from another organism. Or it may have a gene that has been silenced, switched off, so that it's not functioning anymore. So genetically modified organisms have had their genetic makeup artificially changed. For it to be classified a genetically modified organism, that change, so that addition of the gene or the DNA segment, would get passed on to their offspring. Transgenic organisms, TGOs, are GMOs. So all transgenic organisms are genetically modified organisms, but not all genetically modified organisms are transgenic organisms. So to be a transgenic organism, you have specifically had DNA or a foreign gene from a different species added into your genome. So all transgenic organisms are GMOs because they've had their DNA altered in some way, but it's specifically the addition of a foreign gene from a different species. If we're inserting a foreign gene into bacterial cells, as you would know from earlier, we call that bacterial transformation. So an organism that's been transformed is a prokaryotic cell that has had foreign DNA included in its genome. An organism that's been transfected is a eukaryotic cell that has had foreign DNA included into its genome. When we look at genetically modified organisms, generally, more often than not, it's an agricultural crop that's been altered in some way. And what we would do is we would take a particular crop and we would genetically modify it so that it displays a particular characteristic that enhances its rate of production or its survival, increases the yield, increases the productivity we get from that particular crop. So left-hand column of this table shows us some transgenic traits that have been incorporated into some different crops. So we've taken cotton, corn, potato and tomato crops, and we've genetically engineered them to have a gene that gives them pest resistance to a particular pest. We've taken canola, cotton, corn, rice, flax, sugar beet, and we've manipulated its genome to give it a tolerance to a particular herbicide. And you can look at all these other ones and read them yourself, but we've created genetically modified crops that are resistant to viruses, some that have delayed ripening, some that have greater tolerance to things such as drought, flood, salt, altered oil composition. We've manipulated some crops such as rice, golden rice is a good example, to have an increased nutritional value. So if that's your staple food source, you're actually getting more nutritional value from that rice than what you would have if it wasn't genetically modified. And also we can genetically modify things to give them a longer shelf life after they've been harvested, which is obviously a good thing when it's sitting in the shopping centres waiting for us to come and buy it. GM cotton's a good example of genetic modification in Australia. So this was the first genetically modified crop in Australia. So taken cotton plants and inserted a gene from a particular bacterium into the cotton plants that codes for a protein known as Bt. So we call it Bt cotton. And that Bt protein actually gives the cotton resistance against infection by a particular caterpillar. As you can see, 10 years down the track, a second gene was added into that same crop of cotton. That is a gene that encodes for a toxin that defends them against particular insect species. So GM cotton, essentially it's been modified to make it more resistant to attack by caterpillars and by different insect species. Canola plants are another crop that we've genetically modified. So in 2003, we took a gene called the GOX gene from bacteria, and that gene codes for a protein, an enzyme, that breaks down a chemical called glyphosate, which is an ingredient in a herbicide called Roundup. So herbicides are things you spray on plants that kill plants. You use them to try and get rid of weeds. The problem was you can have these weeds that grow in amongst the cotton crops, the canola crops, but the Roundup is also affecting the canola plants themselves. So genetically modified canola plants have this gene to make an enzyme, and when they come in contact with Roundup, they actually take that glyphosate and break it down into something that's harmless. So when they get sprayed, the herbicide kills the weeds and the other plants growing in amongst the crops, but it leaves the canola plants alone. With regulation, so in Australia and in any country really, you need to regulate the use of genetic modification. And we certainly do have foods that have been genetically modified in Australia. You certainly would be purchasing products at the supermarket that have ingredients that have been genetically modified. So typically in Australia, GM foods can't be purchased as fresh foods. They can only be used in processed foods. So you're not really going and buying a genetically modified apple or a genetically modified orange, but you might buy some sort of processed food that uses an ingredient that has been genetically modified. Anything we buy that has been genetically modified does need to be clearly labelled with the words genetically modified. So any foods that contain what we call novel DNA, and novel DNA is DNA in an organism that, due to genetic engineering, is different in its structure to the DNA normally found in that organism if it wasn't genetically modified. So if you read the packages of the foods you buy, if it's using genetically modified ingredients, it will say on it genetically modified, except for the following few exceptions. So really highly refined foods such as oils, margarine, sugars, baked goods. Foods made at bakeries, restaurants and takeaways, so they could be using genetically modified ingredients, but they don't need to tell you that they are. Food from animals that are fed with GM feed. So these animals aren't genetically modified themselves, but they're being fed on a genetically modified type of food. So they would be your three exceptions where you're not being made aware that what you're consuming has been influenced by genetic modification. With genetic modification, it always comes under pretty heavy debate, and people get quite passionate over whether it's a good thing or a bad thing. I'm not going to read through the next few slides because you can read through those yourself to have a bit of an insight as to what some of the issues are. What I will point out though is that we can classify our issues under certain categories. So for example, we have debates based around the ecological issues of genetic modification. We also have debates based around some of the social impacts related to genetic modification. There's debate over the ownership and the intellectual property of genetic modification and genetically modified organisms. Health and safety issues to do with the consumption of genetically modified foods, and cultural issues that also are underpinned by some factors to do with genetic modification.